This webinar is going to cover the major structures associated with the male reproductive system and also discuss their structures as well as their functions. Some concepts such as spermatogenesis and spermiogenesis, those will be covered more in depth in lecture. So this is just some of the things that we will not be covering during lecture, but you'll need to know these for both lecture and lab. So we're going to start with an overview. So the primary male sex organs, or we refer them as the gonads, are the testes. So you'll notice here it's spelled with an I. That is singular for one testis. Most men have two. The plural is spelled with an E. So testes is plural, testis is singular. The testis is responsible for both production of the male gametes, the spermatozoa, as well as the production of the male sex hormones referred to as androgens. So androgens is a term that we use for the male sex hormones. The sperm will be produced within the testis, and then they will travel to the epididymus here. That's the spelling for singular, which is a coiled tube that sits on top of each testis. They mature there. I'm going to go to a sagittal section so you can see a little bit better the transport. So it goes from the testis to the epididymus where they mature. Then it is propelled through the ductus deferens, or also referred to as the vas deferens. So ductus deferens or vas deferens. Males, you have two, one on either side. It's propelled through this muscular tube all the way up and to what we refer to as the ejaculatory duct, and then it goes down through the urethra here that runs through the penis. The sperm will mix with various glandular secretions, in which we'll talk about in a little bit. We'll look at the different secretions from each of the glands and their functions. So this is just a basic overview. So let's go back to look at the testis. So here's an anterior view. So you have two. And you'll notice each testis is held outside of the body, and they're enclosed within a scrotal sac. So each of them is enclosed with this scrotal sac, and they have to be kept outside of the body because the testes must be always about 2 to 3 degrees Celsius cooler than the pelvic cavity, and it's essential for normal spermatogenesis. Now, the testes were formed before birth within the abdominal cavity, and shortly before birth, come here, they actually descended through this structure called the inguinal canal and then descend down into the scrotum. Now, if one or more of the testes fail to descend, what results is what is referred to as cryptorchidism, which literally means, it's from the Greek words for hidden testicle. Now, the problem here is not only is there a higher risk of cancers with boys who do not have their testes descend, or they do not have them surgically move down into the scrotum, but they also will be infertile. So again, comes back to the question is why the testes are outside of the body, because it's very important for normal spermatogenesis that sperm production occurs a little bit lower than what body temperature is, the internal body temperature is. I'm going to go back again to this picture and kind of look at some anatomy and also in the various structures that may be important in this temperature regulation that will be important for normal spermatogenesis. One of the things you'll notice is the testes are, again, enclosed in a scrotal sac, which has some skin. Now, the dartos muscle here is associated with the skin, and its tone gives the scrotum that wrinkled appearance to it, but it'll also be important in temperature regulation. So we're going to come back to that one. So I'll put a star by that. The cremaster muscle, which is here, you'll notice it encases, they showed on this one, encases the testes, and it's actually also part of the spermatic cord that we'll discuss very shortly. The cremaster muscle is skeletal muscle. The dartos muscle is smooth muscle. Now the cremaster muscle will be involved in temperature regulation to keep the testes at that little bit optimal temperature for spermatogenesis. You'll also notice that the inner lining of the cremaster muscle is lined by a parietal layer of something referred to as the tunica vaginalis. The testes is lined with the visceral layer of the tunica vaginalis. In AMP1, we talked about serous membranes, and we talked about parietal and visceral membranes. So you should know the difference between a parietal and a visceral membrane. But you should also understand the function of serous membrane. So this is a serous lining. So the tunica vaginalis is a serous covering that actually originated from the peritoneum. So the peritoneum was a serous membrane that we talked about in that abdominal cavity. So it actually originates from the peritoneum, but remember, it kind of descends down through the inguinal canal and then eventually is positioned into the scrotum. Another structure that will be important in temperature regulation will be this structure here, the tympaniform plexus, which is a network of small veins that are found as part of the spermatic cord. So there's a number of structures that are part of the spermatic cord which have descended through the inguinal canal when that testes descended. So it'll have a testicular artery, it'll have the network of veins, it's going to have lymphatic vessels, it's going to have autonomic nerves, which are going to be important in erections, ejaculations. It has the vas deferens, which is that tube that propels the sperm up, and the cremaster muscle. So that's all part of the spermatic cord. Now let's go back to the role of these ones I'm starring. What are their roles in temperature regulation? But also I want to talk about the skin associated with the scrotum because that will be involved in temperature regulation. So first, the skin of the scrotum is very thin and it does not have a lot of adipose tissue. So remember we did integumentary system and we talked about the layers of the skin. We talked about that subcutaneous layer which has a lot of adipose tissue. Here it doesn't. So having very little adipose tissue is going to not allow it to act as an insulator. So it's not going to have very good insulation, but we don't want it. Also, that scrotal skin has a lot of sweat glands. So if the scrotum gets a little bit too hot, it will be involved in cooling to kind of, again, keep the temperature at its optimal. The dartos muscle, which we said was smooth muscle, it's also involved, it'll be involved in temperature regulation because it affects the surface area that's available for heat loss. So if the muscle is contracted, what happens is it reduces the surface area for heat loss. Thereby, if it does that, it reduces heat loss and then warms the testicles. If that muscle is relaxed, it does the opposite. So when it relaxes, it increases surface area for heat loss, promoting heat loss, and then allowing the testicles to cool. So one would be involved, say, if the environment is cold outside, one will be affected or the opposite will happen if it's warm. Now the cremaster muscle is also going to be important, and the cremaster muscle acts much more quickly. Now this is skeletal muscle that if it's a cold environment, the muscle will contract and actually pulls that testes closer to the body. If it's relaxed, it moves the testes further away from the body, which would be more of a warm environment. It's going to try to keep it at optimal temperature. Most gentlemen are aware of what happens to the scrotum if it's cold out, or if they jump into a cold ocean, or they'll notice that their testes pull up closer to the body, which is very important to maintain optimal temperature. And there's also something referred to as the cremasteric reflex. This is a reflex that physicians can check, in which they just lightly stroke the inside of a male's thigh. And what it does is it triggers a reflex which will pull up the ipsilateral testes, and it's used to test functioning of a nerve that controls the cremaster muscle, which is the genitofemoral nerve. So I'm going to write that out for you. Genitofemoral nerve. Okay. Now the last structure that'll be involved in temperature or local temperature regulation is that pympiniform plexus, which is a network of veins. It's like multiple spermatic veins that come from the back of the testis and parts of the epididymus. And what happens is, because of the arrangement of the plexus relative to the testicular artery, it's arranged in a countercurrent fashion, can't spell, in such that the warm blood from the testicular artery moves and carried away into the blood of the pympiniform plexus, which allows that testes to stay a little bit cooler, because you don't want the testes the same temperature as what was your internal body temperature. So you want it to be just 2 or 3 degrees less than body temperature. Does not seem like a significant difference, but it is. If the temperature of the testes stays elevated just a little bit, the male will be infertile. Now, if we look at the testes in section, you'll see here's the one testes, here's the other. You'll notice that it's divided into lobules, which contain all these coiled, convoluted tubules called the seminiferous tubules. So these seminiferous tubules are the location of where the sperm will be produced. Now these tubules will unite into what we refer to as the root testes, which is here. And then eventually it's going to travel and empty into the epididymus, where we'll talk about a little bit later where they'll mature. Now also on this section, you'll notice again the two layers of the tunica vaginalis that we talked about, the parietal and the visceral layer. But you'll notice this other tunic or layer. Tunic kind of means coat. The tunica albuginea is actually fibrous connective tissue that encloses the testes. So you have a lot of different layers. Most of the time you think there's some sort of encase, help protect the testis. Now the tubules themselves are lined by germinal epitheliums. Here's a cross section through these tubules. And there's 2 types of cells. There's a whole bunch of these spermatogenic cells, which will give rise to your sperm, and we'll talk about this in lecture. Let me go a little bit, another view. They're in various stages of development, and this is something that will be talked about during lecture. You'll also notice within the seminiferous tubules, you'll have these Sertoli cells. They're also referred to as sustentacular cells or nurse cells. I prefer Sertoli cells. And we'll talk about the significance of those. It'll be very important in normal sperm development. What you'll also notice on this picture, you'll see over here and over here, or they have labeled as interstitial cells, but they're also referred to as the interstitial cells of Leydig. Now, the emphasis is they're interstitial cells. They are not within the seminiferous tubules. They're outside of it. These Leydig cells, and again, I prefer Leydig cells. The Leydig cells are going to be important in that they produce the androgens that will be needed for normal sperm development. And we'll talk about the whole process of that during lecture. Here's another view from a histological, an H&E slide of the testes, of the seminiferous tubules. So you see the Leydig cells here. You have these Sertoli cells, which are found within the seminiferous tubules, and you have sperm in various states of development. Now, here is a slide from a male who happens to be infertile. And what you'll notice within these tubules, you really don't see a lot of sperm in various stages of development. What you're seeing is an abundance of Sertoli cells. You're not going to be fertile without those sperm cells. I don't care how many Sertoli cells you have, the sperm cells are going to be needed, are going to be important. So let's kind of talk about, you got the sperm developed, which we'll talk about in lecture. The sperm have these little heads and tails, but the thing is, like with that tail, it's like, oh, they should start swimming. They don't. The sperm, the spermatozoa, which is plural, spermatozoan is singular, the sperm that leaves the testes that then enter the epididymis is not capable of fertilizing the egg. It is not capable of swimming. Even though it may look like it's good to go, it's not. So it has to go into the epididymis, where it's going to be stored and also where it's going to help to mature the cells. So it's going to come into this structure right here. Here's the epididymis. And the epididymis, look at a slide of it, the epididymis is lined by stratified epithelium. And that stratified epithelium contains something called stereocilia. Actually, I'm sorry. The epithelium is pseudostratified, so change that. So we talked about pseudostratified ciliated columnar epithelium in AMP1 when we talked about lining the trachea. Now this is also pseudostratified, but it's not ciliated. It has what we call stereocilia, which is incapable of movement. It looks like cilia, but it's structurally more like microvilli. So if you remember what microvilli do, then you're like, okay, you can kind of figure out what the stereocilia are going to do. Well, the stereocilia are designed for absorbing fluid. So it's involved in absorption of large amount of fluid. So because the sperm are not motile yet, there's a lot of fluid that's secreted along with the sperm into the epididymis because it helps to propel, kind of creates a current to draw them into the epididymis. So therefore then the stereocilia are there to absorb large quantities of fluid. And that process actually is, interestingly enough, the absorption of fluid is estrogen dependent. And estrogens are referred to as the female sex hormones. So normal spermatogenesis maturation still involves estrogens. So this one is actually estrogen dependent. So here is a closer up view where you can see the SC is the stereocilia here. Kind of looks like cilia, but it's not. It's mainly really microvilli. And here's your pseudostratified. So it looks like it's multiple layers, but it's not. It's just one layer. So it's your pseudostratified epithelium. Now the epididymis also has various secretions which will help the sperm mature, be able to get to that ability to be able to be motile, and have the ability to be able to penetrate an egg. Now these secretions are actually androgen dependent. So you have to have proper amount of androgens for that to happen. So I want you to start thinking about, if a male does not produce sufficient amount of androgens, he can be infertile because you need it for normal sperm development and also for maturation. The epididymis also secretes a substance that prevents premature capacitation. We're going to mention what that is very briefly. So premature capacitation. So what happens if the sperm become capacitated too early? So we want the sperm to kind of stay quiet, or we say quiescent, before ejaculation. We don't want premature capacitation. Well, capacitation, oops, sorry, good here. Already did that. Capacitation is a process that should take place within the female reproductive tract. And I kind of like this picture because it kind of makes like, hey, you're getting ready for the date. You got the curlers on and you're kind of getting ready, just because remember the whole job of a sperm is to fertilize an egg. So it has to have the ability to fertilize an egg. And the capacitation, we'll learn about the structure of a sperm. It has the head has this helmet called an acrosome. And you have to start to destabilize that head membrane so then the acrosome can release enzymes to help with the fertilization of the egg. And the secretions from the epididymis kind of keep it quiet. It's not going to get prematurely capacitated, but as then during ejaculation it's going to mix with other fluids, hit the female reproductive tract where it's just the right environment at a certain time that hopefully you'll get capacitation. Now, you don't want capacitation too early because it's followed shortly by something we refer to as hyperactivation. Hyperactivation is when the tail becomes extremely motile. It has very forceful actions, and it's going to allow that sperm to travel to the egg. But you don't want it happen too early because it'll peter out. It'll lose its energy. It will never make its way to the egg. So you don't want the hyperactivation because capacitation is shortly followed immediately by this hyperactivation. So you don't want that to happen prematurely. And what's interesting is marijuana, substance in marijuana, causes premature hyperactivation. So people who smoke marijuana on a consistent basis, it's associated with infertility because the substance causes hyperactivation. So the sperm kind of peter out and never make their way to the egg. Okay, so we've got it. The sperm maturing, staying calm, then a male ejaculates. So it's got to be, the sperm needs to be propelled from the epididymis and parts of the ductus deferens, also referred to as the vas deferens. It's either ductus or vas deferens. And it literally means that the term refers to as a carrying away vessel, vas is vessel, or carrying away duct, ductus. So the deferens means to carrying away because it's going to carry it away. So this is a muscular tube. It's lined with smooth muscle, which in its smooth muscle, it lines it and it's involved in peristaltic actions, peristalsis. So it's going to propel stalls. It's an LSIS, which propel the sperm. And you'll see again we have a sagittal section, so it's going to come up here through the inguinal canal, following up here, and it's going to, that ductus deferens eventually fuses with the ducts, you get kind of missing part of it, of the seminal vesicle, and this is the ejaculatory duct here. And here's a different view so we can look at it, a different section. So here is the vas deferens coming around, and it's here's this one's a better view. There's the seminal vesicle, and it forms a duct called the ejaculatory duct. And the ejaculatory duct's not very long, and it passes through this here, the structure called the prostate gland, and empties into the urethra. Now the urethra in the male, actually I'm gonna erase this to show you the length of the urethra in the male. The urethra in the male goes from here all the way out that way. And it exits at the end called the external urethral orifice or a meatus, okay? Now, the urethra is capable of conveying both sperm and urine, because right here is the bladder. So in males, the urethra carries both. In the females, the urethra only carries urine. The female has the vagina instead of, it actually doesn't convey an egg, but we'll talk about that later. So in the male, the urethra is much longer than in the female. And it's divided into sections depending on where you are. You'll notice the part here that runs through, and I'll put it in green, if I can get it to go green, is the prostatic urethra. Very narrow part, I'll do yellow, is the membranous urethra as it crosses through this urogenital diaphragm. And the longest part, we'll just keep in blue, here is the penile urethra, also referred to as the spongy urethra. Now the epithelium, interestingly enough, of the urethra changes as we move throughout. It starts as transitional epithelium. So you'll learn that in AMP1 that transitional epithelium lines the bladder, aligns the urethra, but now it's like, well, in the male, only a certain part of the urethra. but then it changes as it moves down. By the time it gets down here, it's stratified squamous in the external urethral meatus. Now, here's a question. It's interesting. Is people always realize, or maybe you don't, that females are more prone to having urinary tract infections. It's not as common in males. There's a number of reasons. One of the reasons is virtually the length of it. The urethra is much longer in the male than the female, so the bacteria have a lot further to travel up. Also is, you'll notice, here's the opening here, here's the anus over here. You got all this stuff in between. There's a lot further distance. Girls, it's a lot closer, okay? So that's good. So that's why it's very important when they talk about the women wiping front to back, because the proximity of the anus to the urethral opening is so close. And the other reason why males don't get urinary tract infections as easily as females is because the way this urethra is designed is it comes through, it pretty much runs parallel to the penis, but when it gets down to, let's see if I can get here, down here at this opening, it runs a little bit different. So what it does is it creates a spiral stream of urine, which kind of creates a little bit of turbulence and helps clean it out. So you're less likely to have bacteria that travel up. So men don't often get UTIs, but it's not saying they never do. Now, I want to talk about here, I also want to show you something interesting, is if you're having a surgical procedure, you're in the hospital, sometimes they have to catheterize you. And this is where they take a tube and they run it up the urethra into the bladder so they can empty the bladder. You'll notice in the female, they don't have to go as far. Male, you got a lot further, plus that noticeable bend in the penis it makes it a lot more difficult to catheterize a male than it is a female. Female is much easier to catheterize. And so we've got the sperm traveling from the vas deferens to the ejaculatory duct through the urethra, but it's going to mix with a lot of glandular secretion. So semen itself is usually about maybe 2 to 5 mils of fluid during an ejaculation, contains a lots of sperm. And mixed with the sperm, you have various fluids from the prostate gland. And I want you just so you notice that you have one prostate gland, if you're a male, that encircles the urethra. You've got paired, you got 2 seminal vesicles, see right here. And you also have 2 bulbourethral glands that we'll talk about. So it's going to mix with various secretions. We're going to look at what they do. Now, as it mixes with it, it's going to help some of the things are going to give it energy. It's going to enhance its motility once it hits the female reproductive tract. The sperm is viable longer than the oocyte in the female. And some people would say it can live 72 hours outside, but I think that's super sperm. But it can live up to 48 hours once it's ejaculated. And in the female reproductive tract is where we're going to have that final capacitation. It's going to be able to eventually, hopefully, penetrate the egg and fertilize the egg. So let's just look at some of the secretions. So we have seminal vesicles. So you have a section here of the seminal vesicles. We're going to look at the prostate gland, and we're also going to look at the bulbourethral gland. Now, what you'll notice with all of them, they do all have associated with them is some muscle, smooth muscle, because when the muscle contracts, it's going to help to kind of propel out the contents into the various ducts. So we're going to start, let's do the seminal vesicles first. Actually, here's a picture of the vas deferens, just so you see how muscular it is. Now this seminal vesicle, we'll look at the seminal vesicle, this one. Now the secretions from the seminal vesicle are alkaline. So the pH is a little bit above, to be alkaline, it's got to be above 7. Now the reason why it needs to be alkaline is that it helps to neutralize the tubular fluid contents because of the action of the sperm. They may be producing some acid components. The female reproductive tract tends to be a little bit more acidic, and to prolong the life of the sperm, we need to neutralize that. So it helps to neutralize some of the acidic contents of the female reproductive tract. It also, the fluid contains fructose. Fructose is a monosaccharide sugar that is used for energy in the sperm. People always talk, and I'm one of them, glucose is the king of all sugars, but sperm love the fructose, so that's needed for some energy. You also have prostaglandins. The prostaglandins will aid in contraction of the female reproductive tract on its way for the sperm and the egg to meet. And it also contains something called vesiculase. You'll be able to remember this because vesiculase got vesicle as part of it. Now, the vesiculase causes semen to initially clot when it hits the vagina. So a fresh semen sample will clot. In about 30 or 60 minutes, it will start to liquefy. Now, why does semen clot? Well, it kind of the idea is it traps it up there in the female reproductive tract so it doesn't fall out. And then it also kind of traps in the sperm. After it's liquefied, and we'll talk about what substance allows it to liquefy, then the sperm can start to go on its merry way and travel. Now, and also you'll notice that when they use the term semen, it's so close to this word seminal vesicle. This is because about 60% of the secretions of semen comes from the seminal vesicle. Now, believe it or not, though, the first ejaculate contains mostly secretions from the prostate gland. Now, the prostate gland, the fluid is also alkaline for the same reason that we talked about. It also contains fibrinolysin. Fibrinolysin is something, it's just similar to the thing when you talk about blood clotting, is it liquefies that clotted semen. So then the sperm can start to swim towards the egg. Now, the other secretions I want to talk about is the bulbourethral glands. In the male, they're also referred to as Cowper's glands. Now, these glands will help to they're usually the secretions the first to run down the urethra to help to cleanse the urethra, because you've got to remember it's a passageway for both urine and semen, but it also acts as a lubricant. Now, I want to kind of just go so you can kind of see where each structure is. So let's look at the anatomy again. You saw it here in a front view. So you've got seminal vesicles, you got the prostate, and here's the bulbourethral glands. And let's look at a side view. I don't like that one. So side view, seminal vesicle, prostate, bulbourethral. Okay? Now, last structure I want to talk about is the penis. So the penis is the male excitatory organ. This is what needs to be inserted into the vagina during intercourse. But also remember, it conveys both, besides semen, also urine. Now, it's composed up of 3 columns of erectile tissue. You have a paired corpora cavernosa. Corpora is plural for corpus. Corpus means body. So you've got 2 corpora cavernosa, and you've got a corpus spongiosum. Now, the corpora cavernosa lie right next to each other. You can see it over on here. So you have the corpora cavernosa on each side here. They lie on the dorsal side, and the corpus spongiosum lies between the 2 corpora cavernosa. Now, that is found on the ventral surface of the penis. Okay, so one's on the corpora cavernosa are on the dorsal side, the back, and the ventral is kind of the front of the penis. Now, the autonomic nervous systems, it was discussed in AMP1, and should have been its role in male reproduction should have been covered, and it's definitely involved in erection, and it's the parasympathetic division that's involved in erections, while emission and ejaculation. Now, emission is just the movement of the contents within the male reproductive tract. Once it leaves the reproductive tract, it's referred to as an ejaculation. The sympathetic nervous system is going to be involved in that. So you can see here, here's the corpus spongiosum, which surrounds the urethra, and you've got the corpora cavernosa. So the cavernosa is on the dorsal side, and the corpus spongiosum is on the ventral side.